I'm Dr. K.AMUDHA from Biochemistry department. In today's topic, we can see what is metabolic changes that takes place during diabetes mellitus. So we'll see hyperglycemia. So as the name implies, that is it is increased level of glucose in the body is called as hyperglycemia. And this occurs as a result of decreased and impaired transport of glucose and also impaired uptake of glucose into the muscles and adipose tissue. And this is caused due to repression of the key glycolytic enzymes like glucokinase, phosphofructokinase and pyruvate kinase. And there is also depression in the key gluconeogenetic enzymes like pyruvate carboxylase, phosphoenol pyruvate, carboxykinase, fructose bisphosphatase, which promotes gluconeogenesis in the liver. And there is also elevated in the amino acids in the blood, particularly alanine, which provide fuel for gluconeogenesis in the liver. Amino acid level. The transport and uptake of amino acids in the peripheral tissue is depressed. So there is increase in the circulating amino acid level. So once the synthesis of protein is affected and there is an increase in the breakdown of the proteins are broken down to amino acids. So there is increase in the level of amino acids level. The glucocorticoids increases the catabolic action of the proteins in the peripheral tissue so that there is increase in the release of amino acids in the blood. So once there is increase in the amino acid in the blood, the amino acids in the liver, it is broken down to, that is, it undergoes deamination, and so there is increase in the level of urea, and then the urea is excreted out in the urine. So coming on for the protein synthesis, the protein synthesis is decreased in all the tissues. So there is decrease in the production of the ATP, and this is due to absolute or relative deficiency of the hormone insulin. Next we will see fat metabolism. During fat metabolism, there is decreased in the extra mitochondrial de novo synthesis of fatty acids and triglycerides. And the synthesis is decreased. The acetyl-CoA synthesis also decreased. And from this, the carbohydrate, the ATP, NADP and the glycerol phosphate in all the tissues is decreased. The stored lipid undergoes lipolysis, that is degradation of lipids, and this forms free fatty acids. The free fatty acids interferes with the several steps of carbohydrate phosphorylation in the muscle, which causes hyperglycemia, that is increased level of glucose in the blood. The effects of increased free fatty acids level, which takes place in the liver, inhibits further fatty acid synthesis by feedback inhibition at the acetyl-CoA carboxylase step. Next we will see the effects of increased free fatty acids level. The free fatty acids, which is reaching the liver, the highest concentration inhibits further fatty acid synthesis by feedback inhibition at acetyl-CoA carboxylase steps. The fats are metabolized for energy. So once there is increased free fatty acids, there is increased acetyl-CoA level, and this in turn activates pyruvate carboxylase, and this stimulates the gluconeogenic pathway, which is required for the conversion of amino acid, that is a carbon skeleton, into glucose. The fatty acid also stimulates gluconeogenesis by entering into the TCA cycle by increasing the citrate. The citrate in turn inhibits glycolysis at phosphofructokinase level. And not only this, it also inhibits citrate synthase pyruvate dehydrogenase complex and isocitrate dehydrogenase level in TCA cycle. The acetyl-CoA, which is not used by TCA or for fatty acid synthesis, are diverted to cholesterol synthesis and ketone bodies formation. So there is increased ketone bodies in the blood leads to ketonemia, and once the ketone bodies is increased in the blood, it should be excreted out in the urine, and that is called as ketonuria, and this ketonuria causes acidosis. Effect on glycogen synthesis. The glycogen synthesis is depressed by decreased glycogen synthase activity. So this, by activating phosphorylase, the glycogen through glycogenolysis it is converted to glucose. So there is increased ADP and ATP ratio. So when you want to see other effects of hyperglycemia, and this is due to glycosylation of hemoglobin and formation of glycosylated hemoglobin, that is HbA1c. The HbA1c rises is prolonged and it is uncontrolled during diabetes, that is 3 to 4 times than the normal level. So when you see the non-enzymatic glycosylation of other proteins, the albumin, the collagenase tissues and the Leng's protein such as alpha crystalline increases. So the glycosylation of the collagenous tissues causes thickening and changes in the vessels, and also glomerular basement membrane thickening, and this also causes glycosylation of the Leng's proteins and causes the diabetic cataract. Diabetic cataract biochemically may be due to glycosylation of the Leng's proteins, that is alpha crystalline, and accumulation of sorbitol which produces osmotic pressure. So next, coming on for the sorbitol pathway and phosphoinositol metabolism, long-term diabetes has gradually focused on 3 targets. So one is non-enzymatic glycosylation of the proteins, and second is altered microvascular hemodynamics, and third one is abnormal sorbitol and inositol metabolism. The glucose structure is similar as myoinositol, that is MI. So in the glucose receptors, the myoinositol will come and bind. So there is depletion in the MI and there is increase in the glucose. So this leads to hyperglycemia. The glucose interferes with the myoinositol, that is MI, in many tissues, especially in the intestine, renal blush borders, peripheral nerves and renal glomerulus. So there is increase in the glucose and the myoinositol decreases, and this leads to hyperglycemia. During hyperglycemia, the sorbitol increases. The glucose by the enzyme aldolase reductase, it is converted to sorbitol, and the sorbitol by the enzyme sorbitol dehydrogenase, it is converted to fructose. So once there is increase in the level of the enzyme, that is aldolase reductase, the glucose is converted to sorbitol will occur. So after sorbitol, the sorbitol has to be converted to fructose. So once there is decrease in the enzyme sorbitol dehydrogenase, the conversion of sorbitol to fructose is affected. So there is increased accumulation of the sorbitol. So once there is increased accumulation of the sorbitol, the high millimolar levels of sorbitol accumulates in the lengths of the eye and causes osmotic damage, and this linked to develop diabetic cataract. We'll see the complication of diabetes mellitus. There are 2 complications. One is immediate, another one is late complication. So coming on for the immediate complication, this diabetic causes diabetic ketoacidosis and coma in most of the type 1 diabetes, that is in juvenile onset diabetes, and that is of course the insulin dependent diabetes. So coming on for the late complications, The late complication is due to change in the blood vessels. And there are two types. One is involvement of the large vessel, another one is involvement of the small vessels. So coming on for the involvement of the large vessel, and this causes atherosclerosis and its effects. There is an involvement of the coronary vessels. This can produce myocardial infarctions. And there is also involvement of the cerebral vessels that can produce stroke. So coming on for the small vessel involvement, there is microvascular change, and this also causes thickening of the basement membrane. Diabetic retinopathy. 70% of the diabetic patient will be affected by diabetic retinopathy. And this is a disease of the retina caused by a long-term effects of diabetes. And this leads to progressive damage to the blood vessels in the tissues at the back of the eyes, that is at the back of the retina. So caused due to age at the diagnosis of diabetes and duration of the diabetes, poor metabolic control during hypertension, and also due to nephropathy. So when you want to come for the treatment of diabetic retinopathy, mild cases may be treated with careful diabetic management, whereas advanced cases may require treatment or even surgery. Next is diabetic cataract. 60% of the diabetic patients can cause diabetic cataract. And this is caused due to non-enzymic glycosylation of the lens proteins, that is alpha crystalline. And here the osmotic pressure causes, and this causes a damage and the lens proteins due to accumulation of the sorbitol. The lens of the eye gets oxygen and glucose from the aqueous humor. This is the fluid that fills the front of the eye. During uncontrolled diabetes, the sugar level is increased and causing the lens to swell. Additionally, the lens has an enzyme that converts glucose into sorbitol. The sorbitol collecting in the lens can affect the cells and other proteins, which can make the lens more opaque. So when you come for the treatment for this, the cataracts are removed through 15-minute surgical procedure. Diabetic nephropathy. 50% of the cases will have diabetic nephropathy. Here there is increased blood glucose, and this is filtered by the kidneys. So once more glucose is filtered by the kidney, they may cause damage to the kidneys. And this is characterized by proteinuria, hypertension, edema. The triad is called as Kimmel-Steele-Wilson syndrome. Lesions are often present when syndrome is not developed. And papillary necrosis, that is a dangerous complication, and pyelonephritis is caused. The pyelonephritis is when secondary infections occurs. Next is peripheral neuropathy. The peripheral nervous system connects the nerves from the brain and spinal cord to rest of the body, which includes arms, hands, feet, legs, internal organs and face. The peripheral nephropathy is a disorder that occurs when these nerves malfunction in the sense do not work properly because they are damaged or destroyed. And this is manifested by loss of sensation and tingling. And this is caused due to Mi deficiency. Sometimes there may be associated with the myopathies, that is muscle fibers do not function properly, and weakness of the muscle. Next is diabetic gangrene, and this is caused due to diminished blood supply, that is due to atherosclerotic changes in the blood vessels. And this is also due to hypoxia, that is decreased oxygen supply. So there is a formation of HbA1c and there is a less oxygen capacity. The last one is skin lesions. The patients with the diabetic are easily prone to infections and they can cause boils and even ulcers. There may be necrosis of the skin, that is necrobiosis diabetic corum. they may punctuate depigmentation atrophy. And here, wound healing is delayed. In this topic, we have seen what are all the metabolic changes which happens during diabetes mellitus and the complications of diabetes mellitus. Thank you. Thank you. Thank you.